Monocytes are made in the bone marrow and circulate through the bloodstream all the time. Matter of fact, this is actually a diagram showing the walls of the capillaries, which are the smallest blood vessels, with monocytes going through. If there's a site of infection where foreign organisms are actually making foreign molecules, then the cells which actually make up the walls of the capillary will actually respond and generate their own types of molecules very quickly on their surface. Monocytes coming by are attracted to those, will start rolling and then sticking, and pass either between these cells or even through them into the tissue space where the infection is. At that point, those molecules cause these cells to convert from this sort of spherical circular monocytes to these very extended looking macrophages. And these macrophages are like amoebas, and they actually are your first means of defense. Within an hour or two, they can quickly start attacking and trying to actually engulf and eat and neutralize the foreign invaders. How does the macrophage keep up with very quickly growing bacterial infections? And it does it by switching the way it generates energy. And there are two steps in which this occurs. Now, the first step generates only a couple ATPs from glucose. As it goes around the rest of these steps, converting the glucose into other types of molecules, many more ATPs are generated. So if this produces about one fold of ATP, this produces 20 times more when it goes completely around. The problem is that this step is 200 times faster than this step. And a great analogy to think about it is the automobile. If you had a car that got 20 miles per gallon and had a top speed of 100 miles per hour, then given these kinds of numbers, if you were to make a comparison, then this particular step, if you had only this step, you would only get 1 mile per gallon, so much poorer efficiency, but you now be driving at a maximum speed of 20,000 miles per hour. And that's quite impressive. And that's what happens, the macrophage switches like that. And that's been known for some time. And we have been very interested in that. And what we have done is explored this molecularly, and that's actually the topic of what we've been very interested in our research. What the macrophage does, it actually can fight off cancer. So macrophages can be activated to ward off and fight off cancer. In that process, they actually also will make this switch. The switch is actually different than the cancer cell. It turns out that cancer cells do this because they have mutations in this step 2, which actually shuts it off and only can do this. They're sick. The macrophage, though, is not a sick cell, it's a normal cell. It has, and what we've discovered, are mechanisms that allow this macrophage to, at will, shut this on and off. It becomes a lot like the cancer cell, but it's controllable. It then can attack the cancer cell and do various things that are important to actually try to keep the cancer cell from spreading.